everyone, welcome to mltmcq and notes. In this video, I am discussing about some important points from care and management of laboratory animals based on MLT exams. Common laboratory animals and their scientific names are given here. Rat, its scientific name is Rattus norvegicus. Mouse, Mus musculus. Guinea pig, Cavia porcellus. Rabbit, Oryctolagus cuniculus. Sheep, Ovis aerius. Hamster, Mesochrysatus auratus. Mesochrysatus auratus. Once again, rat, Rattus norvegicus. Mouse, Mus musculus. Guinea pig, Cavia porcellus. Rabbit, Oryctolagus cuniculus. Sheep, Ovis aerius. Hamster, Mesochrysatus auratus. Then next, IACUC. It is an Institutional Animal Care and Use Committee. Institutional Animal Care and Use Committee, which is a standing committee required by federal regulations for most institutions that use animals in research, teaching, and testing. IACUC is a standing committee required by federal regulations for most institutions that use animals in research, teaching, and testing. Animal Welfare Act. Animal Welfare Act signed into a law on August 24, 1966. It is signed into a law on August 24, 1966. Animal Welfare Act is the main federal law in the United States that regulates the treatment of animals in research and exhibition. Animal Welfare Act is a main federal law in the United States which regulates the treatment of animals in research and exhibition. So, IACUC is a Institutional Animal Care and Use Committee. Animal Welfare Act signed into a law on August 24, 1966. Next, bleeding, that is blood collection. Blood collection from animals. 26 gauge needle or size 11 surgical blades are used for blood collection. 26 gauge needle or size 11 surgical blades are used for blood collection from animals. Blood collections are categorized into two: blood collection not requiring anesthesia, blood collection requiring anesthesia. In blood collection, not requiring anesthesia includes saphenous vein and dorsal pedal vein. In the case of guinea pig, saphenous vein is used for blood collection which is not requiring anesthesia. Then dorsal pedal vein and saphenous vein both are used in the case of rat and mice. Then moving to next, blood collection requiring anesthesia, which is local or general anesthesia. First one, jugular vein in the case of rat and mice. Then second one, tail vein, that in the case of mice and rat. Then tail snip, in the case of mice. Then orbital sinus, which is used for rat and mice. Then temporary cannula for rat and mice. Then tarsal vein, which is used for guinea pig. It requires anesthesia. Then blood vessel cannulation for rat and guinea pig. Then marginal ear vein for rabbit, which is very important. In the case of rabbit, we blood collected from marginal ear vein. Then orbital vein, which is also used for rabbit. Blood collection are classified into two: blood collection not requiring anesthesia and blood collection requiring anesthesia. In the case of blood collection not requiring anesthesia, we used saphenous vein and dorsal pedal vein. Then blood collection requiring anesthesia includes jugular vein, in the case of rat and mice, then tail vein for rat and mice, then tail snip for mice, orbital sinus for rat and mice, temporary cannula for rat and mice, then tarsal vein in the case of guinea pig, and blood vessel cannulation rat and guinea pig, then marginal ear vein for rabbit, orbital vein for rabbit. Next, marginal ear vein blood sample collection. The method is commonly used for rabbits. Animal should be placed in a restrainer. What is restrainer? Restrainer is an instrument which prevents the movement of the animal. Okay, then ear cleaned with a 95% alcohol. Size 11 surgical blade or 26 gauge needle used to collect blood from marginal vein. After collection, keep sterile cotton on the collection site and apply finger pressure to stop the bleeding. Okay. Marginal ear vein blood sample collection. This method is commonly used for rabbits. Animal should be placed in a restrainer. Restrainer is an instrument which prevents the movement of animal. Then ear cleaned with a 95% alcohol. Size 11 surgical blade or 26 gauge needle used to collect blood from marginal vein. After collection, keep sterile cotton on the collection site and apply finger pressure to stop the bleeding. Then euthanasia. Euthanasia means mercy killing. Euthanasia classified into primary euthanasia methods and secondary euthanasia methods. Primary euthanasia methods includes carbon dioxide inhalation, inhalation anesthesia, injectable anesthesia. Then secondary euthanasia methods includes decapitation, cervical dislocation. Euthanasia are classified into two primary euthanasia methods and secondary euthanasia methods. Primary euthanasia methods includes carbon dioxide inhalation, inhalation anesthesia, injectable anesthesia. Secondary euthanasia methods includes decapitation, cervical dislocation. Next, disposal methods, which includes rendering. Rendering means mechanical and thermal treatment. Rendering means mechanical and thermal treatment. Then composting, which involves aerobic microbiological decomposition. Aerobic microbiological decomposition. Then incineration. Incineration means waste materials converts into ash, gas and heat. Then fire burning. Then mass burial or open farm burial. Then mounding. Then licensed commercial landfill. Then fermentation. Disposal methods are rendering, composting, incineration, fire burning, mass burial or open farm burial, then mounding, licensed commercial landfill, then fermentation. Scientific names are very important. Rattus norvegicus, mouse, mus musculus, guinea pig, cavia porcellus, rabbit, Horictolagus cuniculus, sheep, Ovis aries, hamster, Mesochrysatus auratus. Then IACUC, Institutional Animal Care and Use Committee, Animal Welfare Act, which signed into a law on August 24, 1966. Then in the case of bleeding, 26 gauge needle or size 11 surgical blades are used for blood collection. Blood collection classified into which requiring anesthesia, not requiring anesthesia. In the case of rabbit, marginal vein is used for blood collection. The procedure we already discussed. Then euthanasia, primary euthanasia methods and secondary euthanasia methods. Then disposal methods. Thank you.